Welcome to homework.
Hello students, the question asked is how is a chloroplast structural protein built, modified and transported to the chloroplast? The process begins in the nucleus where DNA, which contains the genetic information for the chloroplast structural protein, is transcribed into messenger RNA, and the messenger RNA then it leaves nucleus and comes into the cytoplasm where this mRNA is bound by ribosome. The ribosomes then read the mRNA messenger RNA sequence and translate this messenger RNA sequence into a polypeptide chain, and which is the primary structure of the protein. And during this process, a signal peptide is also synthesized at the beginning of polypeptide chain, and as this signal peptide or signal peptide is very important for the localization of this chloroplast structural protein, and this signal peptide directs the protein to this structural protein to the chloroplast. If the ribosomes which synthesize the chloroplast structural protein are attached to rough endoplasmic reticulum, the polypeptide chain, which is the chloroplast structural protein, is traded into the rough endoplasmic reticulum lumen, which is the inside of the endoplasmic reticulum, as it is being synthesized. So during the synthesis of the polypeptide chain, it is translocated or transported into the lumen of the rough endoplasmic reticulum while synthesized. The signal peptide is recognized by a signal recognition particle, which is denoted as SRP, that binds to the peptide and halts translation temporarily. The signal recognition particle ribosome complex then binds to a receptor on the rough endoplasmic reticulum membrane, and translation then resume. The polypeptide chain is fully synthesized and enters the rough endoplasmic reticulum lumen, where it undergoes folding and modification such as glycosylation. The protein is then packaged into a transport vesicle that buds off from the rough endoplasmic reticulum membrane, and the vesicle, this transport vesicle, then carries the protein to the Golgi apparatus. Inside the Golgi, the protein undergoes further modification such as addition or removal of sugar group. Such sugar groups and is sorted and packaged into another vesicle. This vesicle containing the modified chloroplast structural protein is transported to the chloroplast. The vesicle fuses with chloroplast outer membrane, and the protein is imported into the chloroplast. Once inside the chloroplast, the protein is further processed and assembled into the final functional form, becoming an integral part of the chloroplast structure. I hope you found the video useful. Thank you.